At the end of 2019, mysterious viral pneumonia cases were reported to infect thousands of people in the central city of Wuhan, China. The symptoms of infected patients can vary, but mild cases often experience fever, cough, and fatigue. In some cases, it may cause a severe pneumonia, organ failure, even possible death. In January 2020, the central government of China imposed a lockdown in Wuhan and other cities in Hubei to control the outbreak of COVID-19. A highly contagious respiratory coronavirus known as SARS-CoV-2 is confirmed to cause the pandemic of COVID-19. It can easily infect people through oral fecal route. This leads to a huge burden on healthcare system as hospitals are not able to fulfill the demand of COVID-19 infected individuals. To date, there have been reported 7 million confirmed COVID-induced deaths worldwide. In addition, thousands and millions of people lost their job due to the lockdown. Therefore, economic loss also be one of the negative impact of COVID-19. The advancement of global travel network via land, sea, and air provides a route for SARS-CoV-2 to disperse easily, widely, and rapidly beyond control. The novel coronavirus has evolved for 4 months and rapidly spread to other countries worldwide as a global threat. Vaccination is the best prophylactic method to stop the transmission of infectious SARS-CoV-2 among the community. The currently available COVID-19 vaccines are predominantly mRNA vaccine, adenovirus vector vaccine, and inactivated virus vaccine. However, this vaccine platform requires cold chain maintenance throughout manufacturing and transportation, which increases overall vaccine cost. In addition, COVID-19 vaccines are primarily administered via the intramuscular route. The invasive nature of injectable vaccination may contribute to vaccine hesitancy among some individuals. Moreover, conventional vaccine development often requires a relatively long time frame. Therefore, a cold chain free and fast-track vaccine development platform is an attractive solution that can be used for different infectious pathogens by identifying antigens that induce immune responses. Most COVID-19 vaccines are spike-based vaccines. Yet SARS-CoV-2 has been reported to evolve approximately every 3 months, with major mutation occurring in the spike protein. Therefore, reduced vaccine protective efficacy against emerging SARS-CoV-2 variants has been observed. Consequently, both previously infected patients and the vaccinated population may remain at risk of infection from the new SARS-CoV-2 variant. The fast-track DNA vaccine platform represents a promising strategy for combating the rapidly evolving pathogen. In this platform, the immunogenic epitope encoding gene is identified and cloned into an expression plasmid to produce a plasmid DNA vaccine. However, the poor immunogenicity and low transfer efficiency of naked DNA vaccine often result in suboptimal immune responses in vaccine recipients and thereby limiting their clinical application. Therefore, the live attenuated Vibrio chloride strain known as VCUSM14P is proposed as a potential DNA vaccine delivery vehicle. This study aimed to construct a naked COVID-19 DNA vaccine and introduce it into the live attenuated Vibrio chloride VCUSM14P strain as the biological delivery agent. Furthermore, the study sought to develop a room temperature stable Vibrio chloride vector vaccine candidate. Briefly, the SARS-CoV-2 spike gene was cloned into a dual promoter shuttle plasmid. Subsequently, the recombinant plasmid carrying the spike gene was introduced into the VCUSM14P strain by electroporation. Next, expression of the heterologous SARS-CoV-2 spike protein in both recombinant VCUSM14P and transfected mammalian cell was confirmed by Western blot analysis. The vaccine candidate was then formulated using a food-grade buffer and evaluated for long-term storage stability. Lastly, animal study was performed to evaluate the immunogenicity of the vaccine candidate. And how it works. First, the VCUSM14P spike vaccine is drawn by the recipients. The recombinant Vibrio chloride strain will pass through the esophagus and later enter into the stomach and intestine of the recipient. As the enteropathogen, the recombinant VCUSM14P will colonize in the small intestine and further release either spike gene-inserted plasmid or express spike protein. At last, the foreign antigens are recognized by the recipient immune cells and trigger the immunity against SARS-CoV-2 virus. This vaccine platform offers several advantages over conventional vaccines. First, it supports rapid and highly flexible vaccine development with the capability for quick adaptation to emerging infectious pathogens. Second, the intrinsic self-adjuvance property of the bacterial host can enhance the immunogenicity of the DNA vaccine. Third, the vaccine can be delivered via the mucosal route, which induce efficient mucosal immunity to counteract pathogen at their site of entry. Lastly, its stability without refrigeration make it a promising cold chain-free vaccine platform. Thanks for watching. See you!